Okay, hi guys, Iris here. So I'm gonna start off this presentation with DNA extraction. So DNA extraction is basically a lab technique used to extract DNA from a sample. Um, I guess that's pretty self-explanatory. But step one is basically collecting a field sample like we did last Sunday. And then step two is just using a DNA extraction kit. Through like most of the articles, I saw this specific kit being mentioned, the DNeasy tissue and blood extraction kit. And also, when performing a DNA extraction, we should also prepare an extraction negative, which can be like DI water, so we can evaluate for cross contamination. If it turns out like our equipment is contaminated, we can see that in the DI water, then we know to like redo the DNA extraction. It's kind of like a negative control, I guess, a negative control. Okay, next slide. So, steps for DNA extraction. Through my research, I saw that these 4 steps were most consistent with the steps involved in DNA extraction. So step 1 is just a lysis, where the cell and nucleus are broken up. In like eukaryotes, the DNA is like housed in the nucleus. So when it breaks, it releases DNA. It is commonly done through an enzymatic process using the enzyme proteinase K. Step 2 is binding, which basically is when the DNA binds to like a substance, and this can be done through like precipitation or silica binding, but basically what it does is it sticks the DNA onto something. Step 3 is washing, so you need to apply a washing buffer to remove the cell debris and protein, because if we look back to step 1 in lysis, you broke the cell, but there's also a lot of other stuff in the cell besides DNA. So step 3 washing just helps clear the other stuff present in the cell, or like outside of the cell, I guess now, because it's broken, and it removes everything that is not DNA. And step 4 is to eluate the DNA. Basically what this does is it detaches the DNA from the binding substance and dissolves the binding, and now the DNA can be collected on its own. Okay, now I'll be talking about gel electrophoresis. So, gel electrophoresis is a lab technique used to separate molecules such as DNA. Fragments can be matched to each other to identify similar species, or it can be used in conjunction with like a reference ladder, which allows you to identify how large the DNA fragment is. And basically the mechanism behind it is DNA is loaded into these rectangles called wells at the top. They're in a straight row, and once DNA is loaded, it will travel. So it travels from the negative to the positive end, because DNA is negatively charged, and you know that opposites attract. So yeah, the standard way, like I did it before in my classes, were that we would first prepare the gel, which is just agarose mixed with electrophoresis buffer, and then we added slots for wells. And then number 2, molecules such as DNA and RNA are then loaded into these wells, along with an indicator for visibility. And this is useful because as our DNA fragments travel, like obviously we want to see them, right? And number 3, an electric current is applied to allow the DNA fragments to travel. So the smaller DNA fragments travel further because they're lighter, and the bigger DNA fragments travel a lesser distance because they are bigger. Okay, this should be slide 6. So I attached like these images, and I kind of drew arrows between them for like a helpful representation of like I don't know, like, what order to do them in. Like, when I researched this online, like, this seems to be, like, the common order to perform these. So basically, first is DNA extraction. When you extract the DNA from the sample, you purify it so it's just the DNA. And then you perform PCR, which amplifies the DNA fragments so we can study them. Basically, think of it as like photocopying. And then the third step is just like analyzing the DNA fragments through gel electrophoresis. Okay, so this should be slide 8, eDNA analysis techniques. So there are two analysis techniques that we read about, which were qPCR and metabarcoding. I'll start with qPCR. So qPCR stands for quantitative PCR, and it's the same abbreviation as the normal PCR, which is polymerase chain reaction, and it is best suited for single species analysis. And its pros include that it's fast and low effort, it's cost efficient, and it's non-invasive. And its cons are that it does not discern live or dead status, and it loses precision at starting DNA concentrations lower than 1 copy per UL. For the former con, I feel like this can be like reduced, because like we read that eDNA, when it's like in sediments, it means that it's kind of been there for a long time. So maybe that has something to do with like dead species. I don't know, I may be wrong. So as long as we like collect our samples in like the correct way, which is like not scooping all the way to the bottom to touch the sediment, I guess we should be fine. I may be wrong on that though. So, and then the second technique is metabarcoding, which is barcoding based off of DNA to allow for a simultaneous identification of many taxa within the same sample. And this metabarcoding is best suited for community level analysis. And its pros include that it's efficient, it's non-invasive, and there are low prices associated with it. And its cons are that it can lead to misidentification. There may also be database gaps in the reference database, and small specimens may go undetected. And lastly, primer bias and PCR inhibition, which can be reduced through like proper like handling of these procedures. Okay, so this should be slide 9, more on qPCR. For the presenter who's presenting this, in like presenter mode, make sure like you like click the screen, because like I did add a few transitions. Just click the screen when I say like click the screen or something. But yeah, this is basically a more in-depth explanation on what qPCR is. So when I first like heard of qPCR, I thought it was the same as PCR, but they are actually pretty different. So to start off, let's compare qPCR with PCR. So PCR is amplifying DNA. It's essentially photocopying DNA so that you can like amplify the DNA to study and analyze the DNA. And it shows you the results of the amplification at the end. However, in qPCR, it provides results as the DNA is replicating in real time. It is also called real-time real-time PCR. And it can also count the amount of DNA replicated in the PCR process. So click the screen again. You guys should see like this picture that says fluorescent reporter dye, and it should have a Pac-Man as well. So yeah, qPCR uses this process called fluorescence quenching that I'll get to in a bit. But in the meantime, here's a TACMan probe named after Pac-Man. What it is it's basically a single-stranded DNA sequence with an F end and a Q end. The F end you can think of as the fluorescent reporter dye. As you may know, fluorescent means like light, or like something to do with light. So yeah, you guys all know what reporter means, and this will be relevant later on. And the Q end is basically the quencher. The quencher dampens the fluorescence through a process called FRET. So tap the screen again. As you can see on the far left image, whenever the fluorescent reporter dye and the quencher are like intact in an intact TACMAN probe, the F end is dimmed. But in the non-intact probe on the right, the F end is not dimmed. So that tells us that whenever the probe is like broken or like deteriorated, the F dye lights up. That's really important. Remember that for later on. So now let's get to the second image. It should say probe binds to target sequence. So basically the TACMAN probe, as I said before, it's a single-stranded DNA piece, so it binds to another single strand of DNA that has already been denatured. You can see that. It's kind of small image, but try your best to see. And the probe stays there. When it turns to extension mode, when like the DNA polymerase comes to like make codons, like corresponding base pairs for the DNA complementary base pairs like ATCG, AUCG, that kind of stuff, it goes along its path, and it replicates the appropriate section indicated by the primer. And when you see the fourth image, when it kind of runs into the probe, you can kind of see the probe kind of breaks a little bit, and that's how the fluorescent reporter dye, or the F end, it lights up. So that kind of tells us that, hey, yeah, you've reached this point. So I guess now you guys can infer, like, you guys can like take a guess as to how DNA can be, like, the amount of DNA replicated can be like counted now. It can be like quantified. So that is what quantified PCR is. It provides results as the DNA is replicating. Hi guys, so now I'll be presenting a little bit more on metabarcoding, which is another DNA analysis technique. So, as I previously defined, metabarcoding is barcoding based off of DNA to allow for simultaneous identification of many taxa within the same sample. Now, presenter, tap the screen. What does this mean? Tap the screen again, tap the screen again. So you guys should see a sequence of like 4 images here. Try to visualize it to the best of my ability. But as we all know, species are made up of cells. Inside the cell nucleus for eukaryotes, you have chromosomes. Chromosomes have sections of the chromosome called genes, and genes are made up of DNA. A specific region of this DNA is called a barcode, and this is extremely relevant in metabarcoding. So presenter, you can like tap the screen and then tap the screen again. So basically what the barcode region does, it allows us to classify our DNA based on like their taxonomic group. So I'll get to what taxonomic group means in a second, but I guess you guys should know the commonly used barcode regions. So for animals, that is COI or COX1. And these regions are used because they have the barcoding gap, which basically means the same species have less variation than species that are different, which is what we want to identify different species. Okay, presenter, you can tap the screen, then tap the screen again. Okay, now we know what barcoding is. So what's taxa? As you recall, this is my definition for metabarcoding. Presenter, tap the screen 2 times. So taxa is defined as a taxonomic group of any rank according to Oxford Languages. So all of these in the image below, all of these are considered taxa, such as domain, kingdom, phylum, class, order, family, genus, and species. So yeah, it's just a way to classify organisms. And yeah, that's made up barcoding. I hope you guys learned something useful,